Every once in a while, we unlock a potential so powerful it reaches out and changes humanity forever. In 1953, James Watson and Francis Crick identified the double helix structure of DNA from X-ray images taken by Rosalind Franklin. And in the 68 years since, manipulation of the genetic code has exploded into countless applications and industries. Genetically modified crops, mail-order DNA genealogy kits, new vaccines developed in less than a year to combat worldwide pandemics. And still, we have only barely begun to understand the staggering complexity of this tiny but immensely powerful molecule. Within each of us are trillions of identical copies of DNA encapsulated in the nuclei of our cells. Brain cells, liver cells, skin cells all have the exact same blueprints necessary to make every protein our bodies could ever need. But if every cell has the same master plan, why do they all act differently? Epigenetics and DNA methylation. As an embryo develops, it reaches a point where the cells begin to differentiate, to become a heart, a nervous system, a skeleton. Methylation is when methyl groups attach at various points along the DNA strand to either prevent or promote the expression of specific genes. For example, muscle cells don't need the dendrite structures of nerve cells, so those genes are suppressed. Once a cell has been methylated, the children of that cell are usually methylated the same way following each division. But what if they weren't? During mitosis, there's a narrow window of time where each resulting child cell is only half methylated, from the half of the DNA that came from the parent cell. The newly added complementary DNA is unmethylated, but only until new methylase enzymes can attach. Now, what if we could stop the process, or even redirect it? It may be possible to essentially rewrite a cell's fate to become a different cell. The problem is, no one has been able to identify nascent cells at this key cell fate juncture. Current genomic sequencing technology cannot pinpoint this narrow window because their DNA libraries come from cells at varying stages in the cell cycle. That's where I come in. Determining the age of cells based on their position in a biological process is known as determining their pseudotime. No one has yet developed pseudotime based on methylation signatures. My research is to quantify methylation pseudotime in order to identify nascent DNA strands. Once identified, a whole host of new, targeted experiments can be performed with the ultimate goal of redirecting cells to perform different tasks. Someday, from a simple swab inside a patient's cheek, we could grow a replacement liver or kidney, or replenish bone marrow with a perfect match. My work will be a vital step in unlocking this potential, and once we succeed, humanity will be changed forever. Thank you.